I am starting my class. Today I'll be talking about RNA splicing. So we had talked about first we had talked about capping, and we had talked about tailing. After capping and tailing has taken place, we have the next step, which is splicing, or specifically the mRNA splicing. So after transcription in the nucleus, the primary gene transcript, also known as pre-mRNA, is processed by modification and splicing to give a mature mRNA. And this process is RNA splicing. So in this process, there is cutting out of the nucleotide sequence intron from RNA so that the protein coding nucleotide sequence or exons are connected into a continuous mRNA. Is my screen visible?
No, sir.
No, sir.
Is it visible now?
Yes, sir.
Yes, sir.
Okay. So after transcription in the nucleus, the primary gene transcript, pre-mRNA, it is processed by modification and splicing to give a mature mRNA. This process is known as RNA splicing. So involves the cutting out of the nucleotide sequence known as intron from RNA so that the protein coding nucleotides or exons are connected in a continuous mRNA. So you have half exon 1, exon 2, exon 3. In between, you have the introns, intron 1 and intron 2. So this is connected one after the other after splicing. And these introns are removed off. So first you have in the pre-RNA, the cap is already added and a polytail is already added. Then you have these orange color, the deep orange color, saffron color. This is the part of the exon. Amino acid 1 to 30, 31 to 104, 105 to 46. So this region and this region, they are spliced out. They are spliced out and exons are joined together to form a continuous mRNA. mRNA. So this is the process of splicing. 1 to 46, the coding segments have come over here. The mRNA, we know, undergoes a series of modification and processing events before the translation occurs. So the first one, chemical modification, capping and tailing. Second one, splicing, the removal of introns and fusion of exons. And in few species, you see RNA editing takes place. So typically in a mammalian gene, you will have 7 to 8 exons spread. You will have 7 to 8 exons spread over 16 kb length. Exons are short, 100 to 20 base pairs, but the introns are relatively long, so a huge amount chunk is removed off. A typical messenger RNA is 2.2 kb long. So if it is located in the nucleus, the initial pre-mRNA or mRNA transcript is quite long. And that is known as heterogeneous RNA or hnRNA. So RNA polymerase makes the hnRNA. The hnRNA has a variable size and low stability. There are some heterogeneous ribonucleoprotein particles. They are attached to these. So you have uh, pre-mRNAs, hnRNPs, which are added. Some proteins are added. So you can see that the hnRNPs are here. These were little balls. And this thread is the mRNA. They have 4 proteins over here. They are organized as a series of beads. You have seen that you have the cap, tail, intron. These orange colored sequences, they are the introns. They are removed out. In the intron, a special sequence where the intron begins a GU and then you have an AG site. This shows that from here the introns start. They have found out that the first 2 nucleotides of the intron are 5' GU to 3'. This is known as the 5' splice site or donor site. And you have in the end AG. And this is the 5' splice site or acceptor site. And in between, there is a polypyrimidine site that is a track rich in C and U. So just upstream of the polypyrimidine is the region known as branch point, which includes an adenine nucleotide, a branch point. And it is involved in a lariat or loop formation during splicing process. So you have the 3' splice site, polypyrimidine track, branch point A, and 5' splice. This is the structure of a GUAG intron. So there are 2 steps of mRNA splicing pathway. The first is cleavage of the 5' splice site. There is an hydroxyl attack. And it is known as trans-esterification reaction. This hydroxyl group from this adenine, it attacks over here. When it attacks over here, this gets cut and it forms a loop known as lariat. Then this particular region which is freed, this exon, this attacks this region, and this joins, and this loop of intron is removed out. So basically, first step is type 2 trans-esterification reactions which take place. Now there is this is a very simple thing. There is a splicing apparatus. Some proteins are involved in the splicing of GUAG introns. There are this GUAG splicing apparatus. It comprises of small nuclear snRNAs known as snRNAs. These snRNAs, they have been given name U1, U2, U4, U5, U6. They are short molecules which associate with proteins to form small nuclear ribonuclear particles, nuclear protein particles, snRNP or SNRPs. The SNRPs, together with accessory protein, they get attached to the pre-mRNA to form the spliceosome. In the spliceosome, the actual splicing reaction occurs. So, protein snRNP, this is the snRP, RNP. They come and bind over here and form the spliceosome. And this lariat is removed, and this exon 1 and exon 2 are fused together. So there is a committant complex. Once this complex U1 binds to the 5' splice site, and U2AF, it binds to the 3' region. U2 binds to the branch point. And in the pyrimidine rich tract, U2AF65 binds over there. And there are in the ESC region SR proteins which activate U4, 5, U4, U6. And there are HNRNPs which inhibit it, which inhibit splicing. So this is the committant complex. So let us look at this picture on the left. First, U1 SNRP and U2 SNRP RNP come and bind. U1 goes and binds to this region and to the branch point U2. Then U6 and U4 complex and U5 SNRNP, they come and they form the spliceosome complex and do the splicing of mRNA. This is how essentially the splicing mechanism takes place. There is a thing known as alternative splicing. In some genes, for example, there is exon 1, exon 2, exon 3, exon 4, and you have intron 1, intron 2, and intron 3. Sometimes what happens, a permutation combination takes place. Here exon 1, exon 2, and exon 3 are spliced out. Here, this whole region, including exon 3, is totally spliced out. Is totally spliced out. Okay. So as if this exon 3 is also an intron. So from the same gene, you have two different types of proteins being formed. Protein A, which has got exon 1, exon 2, and exon 3, and protein B, which has got exon 1, exon 2, and exon 4. This is alternative splicing. Do you people have got any questions?
No, sir.
No, sir.
Okay. Then I'm ending my class.
Thank you, sir.